And here's our chart. We have a bar chart. The title is Top K-mers. Y-axis is the frequency. X-axis is the K-mer sequences. As we said before, the highest K-mer is the TTG-TT, followed by TG-TTA, and so on. Greetings everyone. In part 1 of our COVID-19 genome analysis, we delved into the genetic data, calculating the composition of adenine, guanine, cytosine, and thymine. We explored GC content and SKU to unravel insights. Now in part 2, we are taking it a step further. Get ready to uncover the molecular weight of our sequence and explore the intriguing world of K-mers. We will not only understand what K-mers are, but also embark on the journey of extracting these critical elements. Now let's get started. Firstly, let's talk about the molecular weight of the genome. We will get the molecular weight of the genome by the molecular weight function. Genome underscore MW equals to we got the genome molecular weight, which is 9,241,219.21 Dalton. The molecular weight is a measure of the mass of a molecule or a collection of molecules. It is typically expressed in Daltons. In this case, the molecular weight refers to the total molecular weight of the entire genome of an organism. The value 9241,219.21 Dalton is a numerical representation of the mass of all molecules that make up the genome. In bioinformatics, the genome of an organism is often represented as a sequence of nucleotides, ATGC for DNA, or AUGC for RNA. To calculate the molecular weight of a genome, each nucleotide is assigned a specific molecular weight based on the elements it contains, which are C, H, O, N, and P. C is the carbon, H is the hydrogen, O oxygen, N nitrogen, and P is phosphate. DNA is composed of repeating units called nucleotides, and each nucleotide consists of a sugar, deoxyribose in DNA, a phosphate group, and one of 4 nitrogen use bases, adenine, thymine, cytosine, or guanine. To calculate the molecular weight of a DNA, we sum the atomic masses of all the atoms in the nucleotides that make up the DNA. The simplified formula to calculate the molecular weight of DNA equal to the number of A's times molecular weight of adenine, plus number of T's times molecular weight of thymine, plus number of C's times the molecular weight of cytosine, and the same for guanine, phosphate, and deoxyribose sugars. The molecular weight reflects the combined weight of all base pairs and the associated molecules that make up the genetic instruction for that organism. Changes in the molecular weight of the genome can have biological implications. For example, mutations or variations in the genome can alter the molecular weight, potentially leading to changes in the organism characteristic or functions. Now let's tell a life story. Imagine you have a long piece of text, and you want to know how often different combinations of letters appear in that text. This information can tell you a lot about the text. That's essentially what K-mer frequencies are. K-mer is just a fancy word for a sequence of K letters or characteristics. For example, if K equal to 3, then ATC, CGA are 3 K-mers. But why K-mer is useful? It helps us find patterns in the text. For instance, if ATG appears many times, it might be an important pattern in the genetic code. In DNA sequencing, we can use K-mer frequencies to find and correct errors in the sequence. When we're studying genomes, understanding K-mer frequencies can help us assemble the genome correctly. In addition, it can also help us spot differences between different organisms' DNA. So K-mer frequencies are like counting how often different small pieces of DNA appear in a sequence. And this count can reveal a lot of important information about the sequence. Now let's calculate and plot K-mer frequencies. Calculate, plot K-mer frequencies. K equals 5. We can change K to our desired K-mer size. And now we sort the K-mer frequencies. And here to display the top 10 most frequent K-mers. And now we create a bar chart for K-mer frequencies. Here we output the frequencies. And here we have it, our K-mer frequencies. As you can see, we have the 5 K-mer frequencies. The most common 5 K-mer is the TTGTT, and we have 102 TTGTT in our sequence, followed by this, which is TGTTA, and we have from it 101, followed by AACAA, and we have 99 of it, followed by TGTTG and TTTTG, and so on, until we get the last frequency, which will be CGCGG, and we have only one in our sequence. And here's our chart. We have a bar chart. The title is Top K-mers. Y-axis is the frequency. X-axis is the K-mer sequences. As we said before, the highest K-mer is the TTGTT, followed by TGTTA, followed by TGTTA, and so on. Now let's explain our code. We choose a K-mer size, as the code started by picking a size for these short DNA sequences, which called K-mers. In this case, it's set to 5, but we can change it to any number we like, and then count K-mers. It then goes through the long DNA sequence and counts how many times each K-mer appears. Imagine we are looking for 5 letters word in a book, and we're counting how many times each word appears. Find the most common K-mers. After counting, the code figures out which K-mers are the most common. It keeps track of the top 10, and then we made a bar chart to help us see which K-mers are the most common. The code creates a bar chart. On a chart, we will see these short DNA sequences, which are the K-mers on the horizontal axis, and how many times they appear on the vertical axis. And finally, we print the results. As the code prints out all the K-mers it found, along with how many times each one appears in the sequence. So when we set here k equal to 5, in this line a variable k is defined and set to 5. This variable represents the desired size of K-mers that we want to analyze. We can change the value of k to any positive integer to analyze K-mers of a different length. And then this line K-mer counts equal to empty dictionary. Here an empty dictionary called K-mers is created. This dictionary will be used to store the counts of each unique K-mer found in the DNA sequence. And then we made a for loop. For I in range len genome sequence dot seq minus k plus 1. Here we set a for loop that will iterate through the DNA sequence. The loop will start at the first position, which is index 0, and continue until it reaches the last validation position where a K-mer of size k can be extracted. It subtracts k from the length of the sequence to ensure that we consider all possible K-mers. And then K-mer equals string genome sequence.seq I to I plus k. Inside the loop, a K-mer is extracted from the DNA sequence. It does this by slicing the sequence from position I to I plus k, which is inclusive. The extracted K-mer is converted to a string for use as a dictionary key. And then here, K-mer counts. K-mer equal to K-mer counts.get K-mer and 0 plus 1 for each extracted K-mer. It updates the K-mer counts dictionary. If the K-mer is already in the dictionary, it increments its count by 1. If the K-mer is not in the dictionary, it initializes its count to 1. In summary, this part of the code sets a specific K-mer size, initializes the dictionary to store K-mer counts, and then iterates through DNA sequence to calculate and store the counts of each unique K-mer of that size, and then sorted K-mers. Here in this line, the dictionary K-mer counts is sorted based on the values, counts of K-mers, in descending order, as K-mer counts items returns a list of key value pairs from the dictionary, and sorted is used to sort this list based on the second element of each pair. The counts indicated by key equal lambda x, 2x, index 1. The reverse true arguments sorts the list in the descending order. So the K-mers with the highest counts come first. Then top K-mers, sorted K-mers 10. Here we created a new list called top K-mers by slicing the first 10 elements from the sorted list of K-mers. And then we created the bar chart. The first one is plot.subplot 2 to 1 to 2. Here a subplot is created within the current figure. The plot.subplot 2 1 2 code specifies that we want to create a subplot grid with 2 rows, 1 column, and we are currently working on the second plot. And then K-mers counts, zip, store, top K-mers. This line we used the zip function to separate the K-mer sequences and their corresponding counts into 2 separate lists, K-mers and counts. And then plot.bar, K-mers counts and color we set it to green. Here a bar chart is created using the plot.bar function. It took K-mers as the x-axis values, K-mer sequences, and counts as the y-axis values, frequency counts of K-mers, and the bars are colored green. And finally, output the frequencies. For K-mer frequency in sorted K-mers, we print the K-mer and the frequency. Here we printed the K-mer frequencies and their counts to the console. It iterates through the sorted list of the K-mers and prints each K-mer sequence along with its frequency count. As we can see, the plot extracted and the top 10 K-mer when k equal 5, indicating the highest K-mer is TTG-TT, followed by TG-TTA, and so on. Thanks for watching. I hope you enjoyed the video. If yes, then please don't forget to subscribe to our channel and hit the like button. Until next time, happy analyzing.